Everyone. Nice to see you. Can you hear me? Oh yeah, okay, great. Great. So we are going to wait for a couple of minutes for the different participants to join us. Um, and today Casey is leading the show. So Casey, please go ahead. Um,
Hi, everyone. I can try and just share my screen in the meantime so we can get the slide up.
what we can do, Casey, I think, is start the presentation of the foundation and the initial slides that we want to show our audience. And this will leave some additional minutes for the different other participants to join in before we actually start the pitches.
Cool, sounds good.
Yes,
Nice to
actually.
see everyone. My name is Cassie. I just joined the 2060 Foundation earlier this year. I'm currently doing a PhD at King's College London, researching cardiac aging, and longevity is one of the very interesting projects for me. So yeah, I'll just do a very brief overview of 2060 Foundation, and then we'll go into the two pitches of Haura and Asima Health, followed by Q&As after each pitch. So yeah, very quickly about 2060. We're dedicated to help defeat aging by the year 2060 through initiatives that unite financial and human capital and by raising public and political awareness. This is one of the flagship projects that we're working on, which is the Longevity Investment Club. And then we have these pitches ongoing every 2 weeks or so, where two startups kind of present their projects in front of a crowd of investors. So we have about 200 members, ranging from investors to researchers and founders. And here is the core team. You have Gabriel, who's the co-founder, and Max. And then we have a range of people here who are contributing to the project. I will put this one up. And then, oh, yes, Brian Kennedy is also one of the kind of main advisory board members. He's one of the key researchers in longevity. So it's very exciting to have him on board on this as well. Putting Hora up for now. And then once you're ready, I can stop sharing and I'll leave it over to Nicholas.
Hi. Um, can I show my screen maybe to show the PDF or?
Yeah, let me just stop sharing here. Yeah, go ahead.
Okay, I will try. So, uh, okay, so now you should see my screen. Perfect. Okay, so nice to meet you, everybody. I'm Nicolas Fritz, the founder of Aura. It's the world first and best, till with the first, but biomimetic fusion multivitamin. That's some kind of buzzword, mysterious word. I'm to explain to you what it means. First, I want to talk to you about the current epidemic of nutrient deficiency that the world is seeing. I know that this foundation is focused on longevity, but the thing is that if you don't have the right nutrients, your enzyme will not work. So you can add a lot of fancy stuff, but if the base is not here, you can go nowhere. And there is a current thinking that nutrient deficiency is very rare, and that's not what the studies show, like you can see on the screen. And another thinking that everybody has is that vitamin, minerals are a plus. You know, something like biotin is good for your hair and just for your hair. But without biotin, you cannot use amino acid. You have entire system in your body that will shut off. And as you can see on your screen, there is, for example, people that doesn't handle, for example, alcohol, and all their life they think that they have a sensitivity, and it's just a deficiency in vitamin B3. So there is a lack of knowledge accessible to the public about every nutrient and every impact it can have on your body, on your life. And every nutrient need other nutrient to work. For example, B5 cannot work just in the natural state. It has to be activated by numerous enzymes that need vitamin and minerals. So when you have one deficiency or two deficiency, it's an entire system in your body that cannot be used. And the issue is, even if you have symptoms, you doesn't feel well, you go to the doctor, you ask to test your blood. They will never test the 28 vital nutrients that everybody needs to just live because it's very expensive. So the majority of insurance company will not cover it. And majority of people will not have enough money to buy extra tests for themselves. So majority of people just kind of walk in the street with no deficiency. And the other thing is that a lot of people think that just eating healthy, organic, gluten-free will be enough. But the issue is that the soil are depleted in minerals. So even if you're going to eat very well, you're going to lack a lot of nutrients. So you're going to tell me, okay, I just need to take some supplements. That's what majority of people are doing right now. But the issue is that if you take, for example, vitamin C in high dosage, you will deplete copper. And even, for example, 400 milligrams of vitamin C is enough to deplete your copper. And that's a dosage that's very usual. So you can really put yourself in a worse position by taking supplements in a hazardous way. And so the real solution is to take a multivitamin that have everything in the right ratio, in the right forms. But what you have on the market right now is either underdosed, overdosed, with a lot of excipient. And sometimes it's very, fancy for biohacker like us, but it's not what you need. So I don't know how much time I have, but
10 minutes. 10 minutes of presentation and then 10 minutes of question and answers.
okay, so 2 minutes. And so with Haura, we saw this problem. And for myself, I had a long story of health issue. I had to kind of biohack myself to health again. And I had to take numerous supplements every day, like 30 supplements per day. And I wanted something that everybody can have access, that's not very expensive, that doesn't have extra unnecessary stuff. And so that's what we've done with Haura, all made in Switzerland. We focus on the TikTok generation, but really everybody can gain from Haura. And so the market of the global dietary supplement is growing very fast. It's one of the biggest market right now. And with Haura, we launched last year. We sold out the first batch of Haura V1, and now we took 1 year to create Haura V2. And yeah, I can show you the team as we're closing. And so currently we are launching our next step in France we're going to do a big marketing campaign. And after that, we're going to focus on America. And so, yeah, it was kind of deconstructed, but I hope that you got the main point. And if you have questions, I'm here.
Thank you, Nicholas. Does anyone have any questions? If not, I can lead with some. Yeah, Gabriel.
I can be the icebreaker. I have one question. It resonates very strongly with me when you say that you need to have the right balance between the different vitamins. And if you take too much of one of them, you could start from an imbalance and go to the opposite imbalance, which is worse. I'm a big proponent of this methodology where you would do blood labs on a regular basis, and you take personalized medicine and vitamins and supplements depending on the kind of doses you have in your blood. Your thing seems to be pretty generic. How is it that is efficient when at the same time you need to highly personalize the different doses depending on the patients and their blood labs?
That's a really good question because we are really going against the market because the market is going in hyper individualization, and I think that's the right thing to do. But currently, even if you do blood test and you take some supplements, for example, you lack vitamin A, you take it. If you don't take all the other nutrients with it, you're going to create an imbalance. And so even if you individualize it, you need a base, kind of your daily insurance that your body got everything it need. And after that, if, for example, you like iron, some people need a lot of iron, you can complement in more of Aura, but that's kind of your base that everybody can benefit from.
I
So
want to follow up on that as well, because how would you know the ratios because it's so individual? So what would you consider like the perfect ratio and how was this established?
We try to be as close to the daily need that everybody need, but it was very difficult in our case because I can show you the product. It looked like this. So it's 1 month dosage. You take every day 1 dosage in water. And the difference, as it's not galenic form, not a supplement that you eat, it's going to be absorbed passively by your kidney very fast. And so the effect is very strong. So we had a lot of nutrients that we had normal dosage, daily no dosage. But as we had this absorption that is very improved and the form, for example, the B12, we don't use just cyanocobalamin, that's the synthetic cheap version. We use methylcobalamin, adenosylcobalamin, hydroxycobalamin, and that's the 3 coenzymes that you have in your body. So we can use very small dosage to have very big effect. And it's funny because even animals need kind of the same dosage of nutrient than us if in a ratio with the size. And so the nutrients are kind of needed in the same ratio for everybody. But some people will need more because they have a deficiency, traumatism, maybe genetic mutation. And we tried to do something that can be tolerated by everybody. For example, the methylfolate that a lot of biohackers love is very, very exciting. It creates a lot of neurotransmitter, and it can be too much for about 30% of the population. So what we do is for methylfolate, we use a very small dosage and other forms that are not too stimulating. So it was maybe 3 years of work to create this perfect balance.
I've got a question. I notice in the chart here that you don't have magnesium nor bioidentical C, which are two critical minerals and elements that your body doesn't produce. You have to take that intake every day, which is excreted out through, your various normal process daily. And I don't mean ascorbic acid, I mean bioidentical vitamin C. And those are critical because they manage the functions of moving the phosphorus into the mitochondria and moving the sodium out of the cell, which is critical in developing energy and giving your body enough go to be able to process some of this. Now, the other question is are these bioidentical food source vitamins, or are they chemistry?
Very good question. Actually, the vitamin that you see on the screen is just some of the vitamin, but the full formulation, I can show you. Maybe I can send it afterward of the website. You can see all the formulation, and we have vitamin C and magnesium and potassium. And actually, for vitamin C, you're right. It's very difficult to find the right version of this vitamin. We choose to use the magnesium ascorbate, so that's magnesium that's reduced with the ascorbic acid to make it less acidic and more bioidentical. And we tried with every nutrient to use natural source, but it was very difficult, even from a compliance. Why? Because, for example, vitamin D3, we had it from lichen, and in France, it's not permitted, and we had to change it for one from animal source. So, yeah, there is a challenge, but we try to have the best for each one.
Another thing, the young lady spoke about ratios. How do I know what my ratios are? Well, you can't get these ratios from a blood test. You have to get it from a hair follicle analysis, HTMA. Do you offer that a direction in your community?
No, that's something that we advise people to do. But the idea is to provide the base to this person, and there is just enough to cover your deficiency if you don't know you have. And even if you don't have deficiency, we have small ratio, so you will not accumulate. So that's something else that we have to focus. Aura is really something that you can take every day, every year. And even if you have a deficiency, it will disappear in 2 or 3 months.
Well, the other thing too, your burn rate, your energy processing rate is different in everybody. So, I think I like what I see, but it's difficult to be able to predict everybody's ratios are going to be the same. For example, sea salt, you get Himalayan salt, ancient salt beds. You have the micronutrients, or I should say the trace minerals, which are critical for processing your cell processing, your rejuvenation, your cell replication, things like that. Or stabilized rice bran, which contains all the bees that you have in your process, which is food. I mean, the best way to get nutrition is through the food. And like you said before, all the food out there, because of all the chemistry that's been used in growing the food, has depleted the value of the micronutrient and the macronutrients that we need daily. And the same thing has to do with animals. So, but I think you're on the right track. But I think that a more explanation on other things that you could bring people into the fold of really how to manage your personal health care through food, through not food conservation, but food intake is really critical because a lot of things counteract, like vitamin C, ascorbic acid really depletes the C in your body. It doesn't add to it because it's a cell guard for your body. So anyway, I appreciate the introduction and the presentation. It was good.
Thank you a lot. Yeah,
You're welcome.
you're right. We have the view about Aura the same as we view food. You know, everybody kind of eats the same food, and people kind of got what they need. Everybody is not in great shape, but they got what they need. And with Aura, it's the same, but you're going to feel a lot better. And it doesn't mean you will not try another supplement in your life, but probably you will keep Aura and find your personal stack. And what we want with Aura at a long-term goal is to be able with AI and home testing to be able to personalize it. But I think the technology are already here, but it's too expensive. I'm speaking about the blood test, home blood test. So that's something that we kind of monitor. But for now, we think that's the best solution that we can bring to the people.
Oh, and another thing, you might add a pH test in the package,
Yeah.
So if everybody knows what their pH is, and if they're accelerating on the alkaline side or the acidic side, they have a way to to recognize that and then neutralize their position to be between the 6, 9, 7, 4.
Yeah, that's a very good idea to be able to track the benefit they get from Aura. And
exactly.
A little other thing is that as we know that we cannot do better than nature, we added some strong natural ingredient like spirulina. And spirulina has a lot of amino acid nutrients that are in synergy. So we try to give really a whole all-in-one experience.
That's pretty hard to do, but I mean, you're on the right track.
Yeah, that's hard.
That's like trying to save. That's like trying to stop aging, but you're on the right track.
That's what we're trying.
Great. Thank you, Nicholas. I think I still have a few questions, but I think in the interest of time, we can send them to you afterwards. And then if anyone is interested, then we can try and connect you with them and see if you can answer some of their questions as well. Thank you so much for the great pitch.
Thank you.
All right. Next up, we have Shalini. Perfect. Who will be talking to us about Asima Health. I'll leave the floor to you.
Thank you, Cassie. Cassie? Oh, yeah, thank you, Cassie. Hi, everyone. So I'm Shalini Gupta. I'm the founder and CEO of Asima Health. Asima Health is challenging or making breakthroughs in the cancer space, which we all know affects the longevity of our aging population. Each one of us has been affected by cancer in one way or another, either through our loved ones or through the disease ourselves. So what Asima is developing is a groundbreaking blood test, which can be used for early cancer detection and monitoring. And in this great endeavor, we have an excellent and highly committed top leadership team, which has experience spanning across technical, biology, clinical research, and commercialization. My own background is PhD in chemical and biomolecular engineering. I quit my tenured faculty position at IIT Delhi in India to start Asima Health in Canada a year and a half ago. And in the past 20 years, I've developed several technologies, some of which have been spun out from my lab and actively published in this space. And I'm also very deeply, driven by this cause because I'm a high-risk individual for ovarian cancer. So my journey follows that of a cancer survivor because my tissue biopsy results came back inconclusive. And I have several cancer survivors in my family. So I've seen that part, and it's growing, I know, at least in India and in the US, and now around the world, cancer is becoming such a big problem. So So with that in mind, we call ourselves a liquid biopsy technique, but whenever you think of liquid biopsy, you think of this big lab with like, multi-million-dollar like sequencers and lots of people. So this liquid biopsy workflow has been so complex, costly, and time consuming that despite being reimbursement eligible, its clinical adoption has only remained to the top 1% or even fewer people. And I'm showing you some of the inefficiencies in this workflow here. This is by means not exhaustive. There's so many more. But just imagine, the physician or the doctor has access to only a few cancer blood tests where there are more than 50 plus cancer types. The existing tests, which are at least in clinical trials or in higher dimension than other tests, are only for a handful of cancer types. Now, when a patient goes to the hospital, they get their blood drawn. Many times they also have to get their tumor tissue that gets sent from the hospital lab. So the hospital lab will coordinate with phlebotomy and pathology to send both these blood tests and tumor tissues to a centralized lab, which is a specialized lab that does this, is the liquid biopsy lab, that performs the test and then will send back results to the hospital or back to the doctor and patient, and then get reimbursed through insurance. Now, the challenges are that, the turnaround time it runs into weeks. The cost of the test is a few $1,000s, and the hospital ends up making no money on these send out tests. In fact, they end up losing revenue because they have to coordinate on the logistics and everything. So this is a major challenge, one of which has really led to the low adoption of this technology. So what Asima has developed is a novel proprietary technology that is much simpler, faster, and affordable. So what we are really doing is shrinking that entire lab operation of liquid biopsy into a benchtop technology as shown here, where we have a fully automated platform technology, which reduces the hands-on time for anybody. This could be an undergraduate, doesn't have to be a specialized personnel to run this end-to-end to give out results in a very affordable and quick way. So doing this solves a lot of the workflow problems that I just mentioned for every stakeholder in the value chain. For example, we are not limited to a few cancer types. We are a pan-cancer test. We require no tissue, so we are blood only. We are meant to be put on benchtops in any kind of CLIA lab or blood testing lab, so that includes hospital labs. So we want hospitals to retain this kind of revenue. We eliminate the entire step of shipping, logistics, and our turnaround time is only a few hours. So it's like 6 to 8 hours from blood collection to outcome. And we come at a fraction of a price. So much so that a person can even pay out of pocket for this test. So how do we do this, right? How are we different from all these liquid biopsy companies we've heard about? So my group has been working on this tool called impedance spectroscopy for a very long time. We started using impedance spectroscopy for colloidal suspensions. We moved on to bacterial drug detection for antimicrobial susceptibility testing. And now we saw how our tool can also be used for cancer detection. So what we do is basically we are measuring the same biomarkers that many of the other companies are measuring, which is the cell-free DNA. It can be isolated from, let's say, a tube of blood. And what we know from the literature is that this DNA, as we age or once we get cancer, it goes through a genome-wide hypomethylation. That means on the whole, our DNA loses methyl groups during cancer. So let's say a healthy DNA would look something like this with lots of methyl groups. A cancer DNA will, on the whole, have less methyl groups. And because of this underlying signature, which is common across, I would say a lot of cancer types, if not all, we are able to induce these nanostructures in these DNA, which look very different for a healthy cancer DNA versus a healthy DNA versus a cancer DNA. And these differences in the nanostructures are what we pick up using our electrode in literally 5 minutes. We get scans that look something like this, where you have the cancer and healthy spectra looking distinctly different. And then we use, actually, all the results I'll show you are without AI/ML, but we can use AI/ML to power this computation to get like a yes/no result, which is what liquid biopsy results currently give you. They give you a yes/no, whether you have cancer or not, kind of qualitative output. So we have a family of trademarks and patents filed on this approach. So I'll tell you what we did. So in India, when this was the test where it was originally developed, I came to Canada as a recent immigrant. The technology was developed through my postdocs and PhD students. And what we found is that when we looked at these 216 patient samples collected from hospitals, we had a good mix of like 50 healthy, 166 cancer types. We were able to detect cancer in 15 plus different cancer types. So think of all top cancers like kidney, lung, colorectal, I don't know, ovarian, brain, head and neck. All of them we were able to detect, and at all stages. So we were able to pick up even stage 1 cancers and with very high sensitivity and specificity. Since then, we have now that we're commercializing this test, we have replicated this result in Canada. So we're focusing now solely on colorectal. So we have done all stages of cancer. We have also tracked patients longitudinally over 24 months, and we are able to get 100% accuracy in their detection, even at stage 1. Our overall sensitivity and specificity is well above 90% in human plasma samples, which if you do a comparison to what exists in the market or what's under development, the brown one here are what the technologies that exist. Liquid biopsy techniques are some of them I'm showing here as representative ones. It's very hard to keep sensitivity and specificity both very high because they move in opposite directions. But we are unique in that sense that our overall sensitivity and specificity over 275 human samples is well above 90%, which is very hard to do. So we're very excited with this sort of preliminary promising data. Now, our business model is very different from the liquid biopsy companies that are operating in the space because we are highly decentralized. We want to sell directly to labs rather than performing our tests. And our labs are hospital labs in North America, and we want to obviously expand to other locations as well. The reason why hospital labs is because they carry majority of the diagnostic test business, even though they're smaller in number, their pipeline of customers is huge. So what Asima will do is, sell the device, which is like a one-time sale, pretty standard razor blade model. We give the device at one-time cost, but then make money off of the consumables. So the consumables will have, the liquid reagents as well as our cartridge. And we plan to sell this to the hospital lab at $200 per test. And the hospital lab can then get reimbursed either through a self-paying customer or through insurance at $350 per test. This is for the consumable part of the kit. So our initial entry focus for the FDA pathway is looking at colorectal cancer, but looking at the diagnostic and the monitoring aspect of it. So I'm just showing you here on the right-hand side how the cancer care continuum really looks like. People are there are tests that exist for screening. That means in asymptomatic people, you can get screening tests like Cologuard. Some of you may have used it. There are diagnostic tests available for ailing patients who come to a doctor with some symptoms. Therapy selection tests, minimal residual disease. That means to see the efficacy of a drug, how you are responding to a drug if you're a cancer patient under treatment. And then once you're in remission, you have to be constantly monitored. So all of these different conditions require testing, and there exists some market for each of them, except diagnosis. I would say there's not a lot of blood tests available here. So we want to enter the monitoring space. That means the recurrence monitoring or the surveillance space for people who already have past history of cancer. And then diagnostic space where there's literally no competition. And we know that the market is large, at least $50 billion or more. And by entering these two phases, we expect a class 2 pathway, which is a lower, like a lower classification than a PMA. So the product roadmap is that with phase 1, we want to start with, like I said, diagnosis and monitoring of colorectal cancer. We want to quickly expand this into other indications. The next one will be lung. And we don't want to stop at these two conditions. I mean, we want to become a screening test, an MRD test as well. One thing I would like to point out is that we are unique in the sense that we require no additional R&D investment beyond the first product that we make, because our core technology, backend technology, does not change from cancer type to cancer type or indication to indication. We're also a gateway to a lot of rich inaccessible genomic data that we have already started building our AI/ML models on. So the hope is that we should be able to increase the predictive accuracy, be able to do even cancer staging. This is something we're filing a patent on next month, and do risk stratification to triage patients into getting, let's say, a colonoscopy or a mammography or whatnot. Yeah, so this is just the timeline. We started our journey in June 2023. And since then, we have done a lot of analytical validation, preclinical validation. This is already 275 now. We have filed our IP and trademarks. We have our first prototype that we are testing. We are also publishing our second manuscript now next month. And we're also doing an AI-based cancer staging AI model. The next steps are I'm raising $500,000. Just want to say that this has been done with very little money. I've only raised $500,000 US. We are very cognizant and very, very careful with how we spend money. Now we are going to raise a 2.5 million USD round for basically making a design logged product, talking to the FDA, getting the first two customers. Basically, I want first two customers to come in even before the FDA so that we can start revenue early. That is projected in the next 2 years, so this year and next, so by end of 2026. And then eventually we want to go for FDA submission as well for colorectal, and that would be somewhere in 2028. So the revenue projection, and this is a conservative view of revenue projection, is I hope to be able to sell about 4,000 tests by 2027. I'm actually pushing this even earlier, and I can talk more about that. And then eventually we want to grow. IVD clearance for colorectal cancer is expected somewhere around 2029. And then from there on, this can be used as a predicate for all our future clinical indications. Again, no cost for R&D investment. It's only clinical validation on a new cohort is what we'll have to do. So yeah, this is the use of proceeds. The money that I'm raising will take me ahead in 4 aspects. One is the regulatory path progress. Second is product development, the hardware development. We have outsourced this. In 3 months, we have our prototype ready. I'm very excited to share some work on that as well. Getting clinical research partners to now test our technology in their labs. That's what we would love to have, like put a device in their lab and for them to start collecting data, and then getting those early 1 to 2 customer contracts. We already have, yes, there's lots going on in this. We're two interesting parties right now we're talking to. And yeah, I would just like to stop here, and I would love for all of you to be part of Asima's journey, and happy to take questions.
Thank you so much, Shalini. That was great. If anyone has any questions, feel free to just unmute and ask it. If not, I have a few I can start with.
Is your product now marketed in the United States?
No, not yet. But I'm looking for, so there are ways you can market without getting FDA clearance through the LDT model. I'm looking at longevity partners. I'm hoping by end of this year, I'll have 1 customer who can do this in their lab setting. So the short answer is no. I don't have any customers as of now, or it's not selling as of now.
Where is this product made at?
Where is it made at?
Yeah, where's it being made at?
So we're using off-the-shelf reagents. For the liquid reagents are all off the shelf, that we buy and we package them together. And the instrument, the first prototype, the one that I showed you here, we have engaged a company in San Diego who are doing all our engineering design, because we have a lean team of only 3 people, but we outsource this to get this device platform made for assay preparation. So this is semi-automated. Like I show 3 functions here, right? Like doing.
So you said it's made in San Diego, California?
The prototype. There's no manufacturing step. We're too early for manufacturing as of now, but yeah, the prototypes are made in San Diego. That's right.
So it would be considered a US product.
Yes, I mean, it's marketed. It's going to be marketed for the US. Canada has a very small market. It's healthcare. It's a little bit complicated here. Everything has been designed for US.
Have you approached the Veterans Administration in the United States for military veterans?
I have not.
VA hospitals?
I have not, but if this is something really interesting, I would love to talk more about this, like the Veterinary Association, right? I've not done that yet. No.
Sure. Well, yeah, that might be an avenue we might be interested in, because we're a government contractor.
That'd be really great, because I think we are perhaps I've done so much landscape work on this, trying to understand this. I think the challenge is most of the companies at this point are failing and sort of over projecting their results. I don't know if you know about Natera's Signatera test, which is one of the more common tests now, at least available through certain hospitals, through certain physicians. And many times they have to redraw samples because the tissue that they find, they have to have a tissue for baselining their blood tests. So they'll do sequential tests. And many times their blood doesn't have the required amount of tissue that you require. They cannot do detection at stage 1. We can pick up stage 1 detection, and we can even track patients to get recurrence, how they're responding to therapy. We have all that data with 100% accuracy, stage 1, stage 2, stage 3. So we're very excited about these results in general. Yeah, go ahead.
You have a patent on this or patent pending or?
We have filed, we have filed patents. They're still pending. They're getting examined, like they've not gone out for examination right now. So we have 3 patents.
Okay, and it's a U.S. patent also? It's protected in the United States?
Yes, yes.
Okay. Yeah, well, I mean, if it does what it says it does, it makes it a lot cheaper, and it cuts down the costs dramatically, especially in the military. In the United States government now is all about cost cutting. So this could be a unique entry into and the analytical side in that type of anomaly, So yeah, I'll put my name and my email, how to get a hold of me in your chat box.
That'd be so great. I would love to follow up.
All right, thank you.
Could you give a sense as to how you see competing against the Grail Galleri test, which has had such success in the US market? I believe it's currently priced at a B2C retail price of $750 per test, and I've heard thoughts that the price could go down further. I wasn't sure if through your market research you have a sense as to just how low they can go, and if your differentiator of price will continue to be to your benefit when you go to launch in the US market. Thank you.
Thanks. Yeah, Judy, I think a couple of things here. So I believe the test for a gallery test cost is about $949. But Grail being Grail, they're highly subsidizing the cost, right? So I think cost is only one of the factors. Another thing is that if you think about this, they are a screening test. So Grail, for example, is launching in the screening market. This market has been hard to get reimbursement for, number one. There are a couple of FDA approvals, 2 FDA approved tests. Grail's test is not FDA approved. The first guy who got FDA approval, Theo, he's now on our advisory board. They got the first FDA approval in 2016. The second one for colorectal cancer has come in only last year by Guardant. Now, saying all of this, the performance of Gallery's test, if you look at this, I've mentioned this here, it's 99.5% specificity and it's very low sensitivity. And any company, if I show you this will make it very clear. Any company which is getting data in healthy and cancer, they have always some overlapping data, right? Because the test, ideally you would like something like this for any test where your healthy and cancer are completely separated, but that never happens. So what companies do is they tune their thresholds in such a way depending on the clinical application they want to launch in. So what Gallery, for example, has done, they have their threshold somewhere here where their healthy data looks very nice, 99.5%, but their sensitivity completely sucks. If you look at this data, this is 51%, right? If you're launching as a recurrence monitoring test, such as us, we would like sensitivity to be very high. So for healthy people, you don't want to overdiagnose a healthy population. Prevalence is very low. So people have like high specificity, but this is suffering. For us as a recurrence monitoring test, we want to be somewhere here. Okay, so right now we are here. I've just said, okay, both of these things are high. Sensitivity, we don't want to miss out on any cancer patient. So how you tune your thresholds completely dictates where you want to go. I don't want to launch as a screening test unless it's for the longevity world where there's a self-paying customer, because reimbursement is hard. FDA kind of comes down hard on these multi-cancer tests, and we don't have specificity to say which cancer type. But we are very well placed to start with as a recurrence monitoring test. So I'm seeing GRAIL as a competitor in the sense of the technology, because they're using cell-free DNA, they're looking at methylation. They're one of the only companies that are doing whole genome. Well, not anymore, but their phase 1 had whole genome because they can do it. They're Illumina's baby, right? So they could do it. But it's very cost prohibitive for any other company. We are doing whole genome data collection in a very unique way, and I can talk more about that. So we are competing head on head with GRAIL, but I don't think GRAIL and we are competing in the same market as of now. Also, minimum cost. So this guy, I'll tell you, this advisor, John here, he was the lead clinical strategy advisor, senior medical director at GRAIL. So was Vidya. She led their commercialization strategy. All of them can tell you that the failure rates are very high. Minimum cost right now is $650 to just get the test. But I'm saying it's not, cost is never the only differentiator, right? Like it's always like a bunch of things that I can give you 21 reasons across operations, business, and clinical performance, but in which we are better than these kinds of companies. And that's something I can discuss offline.
Thank you. That was super helpful.
Shalini, I was wondering if you have any indication that cell-free DNA methylation changes during aging, whether it's specific sites on the DNA, and can you detect it in specific organs?
So during aging, this kind of behavior is seen, right? So if you age, the methylation, overall methylation, when I'm saying like whole genome methylation, it goes down. But in cancer, this goes down like this, right? So the change is just so much higher. It's almost like a step function. So with our test, we do see a slight change with age. Like if I take just the healthy folks and I look at the slope of signal, which is reflective of the methylation, we do see a change. But our assay doesn't have the sensitivity yet to say, look, this is the biological age versus chronological age. We are not pitching for that. We can make it that. It's definitely leading to that. What a True Diagnostic, which is, let's say, a leading company in this space of longevity and methylation analysis for DNA, what they do is they have specialized regions in their DNA, like targeted gene panels that they're looking at, which have known to show correlation to aging. But they, again, are like a ship test. You have to like send it to a centralized lab in, I think, Kentucky. They'll do this test, give you results back 2 to 3 weeks later. We are not competing per se in that space. We could. The focus is not, but we are picking up whole methylation change across the DNA, which is very well reported in the literature. So this statement, genome-wide hypomethylation changes in cell-free DNA that occur during cancer across cancer types, this is a very well-known statement. We're just using it. We're not saying, we're not proving this. It's not our discovery. Our discovery is only to say, look, how can we measure these changes? So if somebody is using a gene sequencer to measure this over like a 2-week period, we are able to do this in 6 hours because we're not looking at individual beads in a necklace. We're looking at the shape in the change in shape of the necklace rather than individual beads. We don't care where that bead has changed, which is what, let's say, other companies which are doing sequencing will look at. We are saying it doesn't matter where the bead in the 3 billion base pair, the DNA changes, it's still going to lead to some kind of a structural change at the bead level, at the necklace level. And that's okay because we can pick that up. We don't have the granularity to say which bead changed, so which cancer, like what's a tumor of origin. We cannot give you that information with our test. As of now, we cannot.
And do you anticipate that an intervention, that these changes are dynamic enough to kind of pick up on interventions? Because I would imagine that for aging, this is what mainly people would be interested in measuring.
Yes, well, let me tell you this. I'll show you some like unpublished data that I really, really like. I told you about longitudinal data. This is data that we are now publishing, but let me just, how do I zoom it out? Just give me 1 minute. I don't know if I can zoom this thing.
There's a little cursor at the bottom where it says
Yes.
204%.
Actually, yeah.
Bottom right corner.
Yeah,
Right,
no, I'm just
right corner.
Yeah, no, I'm just struggling with the Zoom link showing at the bottom. Hold on, just give me 1 minute. Yeah. So if you look at this data, okay, this is not even like this is data that we got for colorectal cancer patients just literally last week, where we have stage 1, stage 2 colorectal, like these are all patients which had a starting cancer So these patients have been tracked over 24 months. Okay, so we bought these samples from a biobank. So these have been collected over 24 months for all these 5 patients. And stage 1, stage 2, this is also stage 2 colon, this is stage 3 rectal, stage 3 rectal. We were able to pick up changes because you asked that. So we're able to pick up changes in states of this. So what you see here, for example, the patient was declared cancer-free. And this is our threshold to say somebody is cancer-free. Like everything above, if you see a signal, the person should be cancer-free. And below, we say it's cancer, right? So this is what we are measuring. And you can see that we were able to track these events. Somebody had cancer, somebody became cancer-free, and then they again had cancer recurrence. We were able to track that. So the person would have had surgery or some therapy here in the 6 months. Here again, we are able to track somebody. So somebody started with cancer and then became cancer-free. We were able to track that with our test in just 6 to 8 hours. Here, the first data point is missing. This person, like time t equal to 0, would have had cancer. We're testing this now. They became cancer-free and continued to become cancer-free over 16 or 13 months, whatever this time point is, and so on and so forth. So we are able to track these changes going from cancer to cancer-free in a highly quantitative way. But right now, there are so many confounding factors per person, right? Because age, like the type of cancer they have, the stage they have, the biology is very different. So if you give me a cancer right now, I cannot tell you what stage it is because of all these confounding factors. But at a population level, I can map out sensitivity changes. Like at stage 1, sensitivity is about 90% or 88%. As I go higher, that's hitting 96%, 97%, which at population level makes complete sense. So we can track, but if somebody has a baseline already, like the starting point, we're able to track them with 100% accuracy. The data is small, like we have only 5 patients at this point because getting these kind of longitudinal samples from biobanks is very difficult. Like people are not collecting these samples many times. And one difference is most of these companies are using Streck tubes, which are those expensive tubes with preservatives. We use standard purple top tubes, the one that you and I probably have used during phlebotomy, because we are meant to be close to the patient. So sometimes biobanks don't have even those because companies are not even using or focusing on those kinds of tubes. So getting the clinical samples to test has been one of our challenges. We are working on this in 2 ways, setting up partnerships with labs, which have a pipeline of folks coming in. And then we're also making our hardware such that it can be placed in that lab to get high throughput data collection. So that's something we're working on at this point.
Thank you. Does anyone else have any other questions?
Yes, I do. So I understand your technology is able to detect cancer with a high specificity and high sensitivity, but is it able to detect what kind of cancer the patient has?
No.
So you have a cancer, you have like 95% of having it if your test generates a positive result, but you still don't know what it is. And what would be the next step when you find out that you have a cancer, but you want to investigate further?
Right. So if I were to go and tell FDA this, they would not like it for a screening test, right? They don't like this multi-cancer test. But because I'm launching myself in 2 areas, longevity, and I'll come back to that because it's so relevant for this group. So I have 2 tracks. One is like this quick revenue, fast revenue track for the LDT, and the second is the FDA track, which is I have to be very careful on where I go with that. For the FDA track, I'm going with recurrence monitoring. So imagine if I am a cancer survivor, and right, so you understand the track for that, right? Okay, great. So the next path would be for a cancer survivor, if they test positive on my test, the patient will go for standard of care, could be mammography, could be a CT scan, whatever it is. But my question is always like, if there is a physician who tests somebody like a breast cancer survivor, they pick up the cancer in mammography, what is their next step? Their next step is they will still go for a full body MRI to see if the cancer has spread, which may be the downward path for us also. For longevity, I think people are okay to pay for full body scans because these people are early adopters. They love to get to know about their health. FDA doesn't like it because it's a huge cost for the healthcare. So they probably would want to have like a follow-on test. So I'm not sure things are changing in the FDA as new companies and liquid biopsy companies are coming forth. But I think for recurrence monitoring space, we're really, really well placed. Also, cost of clinical trials goes down humongously because we are looking at a prevalence rate of 20%. So you have to screen out a smaller population to say your test works versus tens of thousands of patients, because if your prevalence rate is like 0.5% or 1% in healthy population, you have to present the FDA with 10,000 plus data to say, look, I have 50 positive events. Whereas if you do it in a high prevalence risk rate, it's smaller clinical trial costs. I feel also the adoption is nice. You can have a great technology, but if a person doesn't want to use it, you can't push it on somebody. But people who are cancer survivors, they want to take care of their health, right? Because they've been through this once. So multiple reasons, business, commercial reasons why I'm going with recurrence monitoring. Say screening is a PMA, it's a class 3. Recurrence monitoring is a class 2. So that's also like FDA path slightly changes. But the longevity space, I definitely want to have this because it's not non-FDA, it's not regulated. And I've already inbound interest from labs that they want me to put their test in their lab. I visited Arkansas. Arkansas has multiple labs. They want to partner with us, but our hardware is not ready yet, right? Like we're working on that, and I need money to make it robust enough I can start keeping it there. So yeah, some of those reasons why I'm looking for investment.
I have two additional questions. We still have 3 minutes. So one of them is, we all have cancer all the time. I mean, small cancers that our immune system is fighting against very effectively. So there is no point in applying any treatment at all. How do you make the difference between the big cancers that we really need to focus on and the ones that each one of us has all the time and there's no problem? So that's the first question. And second question, from the point of view of longevity enthusiasts, which I happen to be one of them, is it imaginable that at some point, I mean, the prices you announce are absolutely affordable for people, not even labs. I mean, would it be imaginable at some point that I would buy such a device and I would buy your consumables and I would do it every now and then, maybe 6 months or anything? I mean, the price is absolutely affordable.
Yeah.
At
Okay.
the individual level, I mean, yes.
Yeah. So I'll come back to the first question. If you have like some local mutations that the body is taking care of, your body is not producing cell-free DNA, enough DNA that gets spilled over into the bloodstream. So the reason why the blood, like even healthy folks, okay, so let's say if I don't have cancer, all my cells in the body, they're undergoing a turnover, right? They're dying, maybe not at a very high rate. That creates a lot of debris, which goes into the bloodstream, which is what we pick up. So if I just pick up blood from a healthy person, I'll still get some baseline of cell-free DNA concentration, which is coming from healthy organs. When there is a tumor in the body, that turnover becomes so high that the spillage over and above the background becomes higher. Okay, so people, that's the circulating tumor DNA part of it, the ctDNA across cell-free DNA. If there are small mutations happening, that's not going to affect the population of the cell-free DNA because it's just a local event. There's no sort of tumor which has come, which is releasing a lot of cell-free DNA that you're picking up. So we won't, or we as in no company can pick up that early, like local mutations, and you don't want to even pick them up because they're like, there's no information to be, there's no clinical utility of that, right? Body is fighting it, it's all fine. So does that answer your first question about the...
Yes, it does. Absolutely. Thanks for the clarification. Yes.
Yeah, so that will never happen. So the second question about I mean, it's a $5,000 equipment. Would you want to keep a bulky equipment in your house? I don't know. I mean, I've never thought of it
Well,
that way,
honestly, I know people. I would certainly do if
okay.
it's reliable. I know lots of people who would do this. And if you've had family history of cancer, which I happen to have, this would be an additional reason for people who have some money to invest in this. I mean, when you spend $200,000 for a car, you could purchase a 5,000 device to live longer than your parents.
Yeah. No, I hear you. Only challenge is that what you put in the device will not be blood. I mean, we are thinking from plasma. So blood, once you collect, it's a phlebotomy blood. It's not a prick test, number one. So first of all, you need a phlebotomist, right? So it's peripheral blood. Second thing is that blood to plasma transition usually requires a centrifuge. It's a double spun plasma centrifuge, plus you require pipettes. So it's not designed to be that, but I'm not saying it cannot be. Just the vision that I have is more for a lab where one instrument is used as high throughput for like multiple test volumes and where people, but otherwise, once the plasma is in, the hope is that we would not require anything else beyond that point, right? Everything just kind of manages, but you require tubes that can also be managed through consumables. So short answer is perhaps yes, it's a bit of an
Overkill. I see.
Oh, she just froze for
Yes,
me.
I thought it was me, but yeah, we lost it.
Oh,
Oh
can you
yes,
hear me?
we got you back. Yes, you're back. You're
Sorry,
back.
could be my internet. But I'm not saying no, but that would require a little bit more
I
thought
understand.
and yeah, it can be done, but I never thought of it that way. Mostly at a physician's office, perhaps yes. If you can get blood to plasma transition, usually it's done through centrifuges, which are portable centrifuges are available nowadays. I think those are also possible to get. Okay, but I'll think more about this,
Yeah, it's
Gabriel.
just a question, just an idea. Thinking of myself and people around me who have had cancer themselves or have relatives who've had this and are really anxious about this, understandably. Can we sign up for your clinical trials? Do you have this? How do you get the data? Would it be helpful for more like patients to onboard on this?
Yes, so I've been getting a lot of inbound requests. The challenge is, I'll tell you, because most of the current companies, they do a blood collection through a Streck tube, which keeps the blood okay without any spinning for 7 to 10 days. We have never tested in those expensive tubes because we're such a low cost test close to the patient. If you had to send us your sample, you would have to get it extracted and send it on dry ice because the plasma cannot stay without like cold conditions. It requires a cold chain. So I am figuring out, maybe I should just test on Streck tubes and see if the data is the same. I have never done that because it was always done on those purple top tubes. So yes and no. But if you have a pool of people, let's say you have enough, because dry ice costs about like $250. It just increases the price of a test. You have to buy dry ice, go to FedEx. Like it's just a little bit of that. But again, the answer is right now I'm not ready, but at some point I will be, right? Because this is something, if I there's enough interest, I can find some ways to get the data. So let me get your information. I mean, I have your information, Gabriel. Like I don't need your
Yes.
information, but I
Yes.
will reach out to you. Let me just put it this way. I'll reach out to you to get your blood in some way or another to be able to run it at our facility for sure.
Yeah, it would be interesting as an experience, and also yes, to get an eventual answer. Yes.
Okay, sure. I'll find a way to, I'll get back to you on that. I just have to think of innovative ways to
And
get your blood.
to use my example with other people who I happen to talk to every day and who have had again family history in this diseases category of diseases, and who are really careful at this chronic disease.
Yeah. No, absolutely. And I am the same. I have five cancer survivors, one cancer death in my family. I'm a high risk. So I know exactly what you mean. And I want this test to be out in the market. One thing I did not mention is that other liquid biopsy companies, they're so reliant on AI. Everything that I showed you has no AI, no bioinformatics. It's literally you measure, that's the raw data you get. Our path to market is very, very quick. Okay, so where we are, I mean, we need money to be where we have to be, but by end of this year, we can make this test accessible through labs to multiple people. If there is a local lab, we can keep this in. This should be able, like we should be able to do it as a longevity test for sure. So if not now, but definitely by end of the year, we'll have an answer for you for this part.
Okay, thank you.
Yeah.
I mean, time's running out, so I don't want to ask any more questions, even though the topic is really fascinating and promising.
Happy to chat afterwards if there's interest. I'm happy to connect one-on-one with
Okay.
anyone.
I have a
Yes.
few questions I'd like to pass on as well, but I think we can end it here for today's session.
Sure. Okay.
Thank you so much, Shalini, and everyone for attending.
Bye.
Thank you, everyone. Bye-bye.
Bye, everyone.